Thanks a lot for the invitation and for this kind introduction. Thanks, Antonio, for organizing this great conference. So today I will try to give you a brief overview about some of our main works of the last years, where our main goals is to try to identify preserved brain functions in altered state of consciousness, and specifically sleep, but also coma, and how this information relate to the long-term outcome of these patients. We're interested in identifying this underlying neuromechanism and to understand what are the features that are common across varieties of unconscious states. And also more recently, we're looking into the interaction between interoceptive and exteroceptive signals in this altered state of consciousness. But before starting, I would like to give you a little brief overview about the context of these studies, and also to introduce this quite elusive concept of consciousness. So for a long time, this underlying neural mechanism that allow us to be conscious in different setting has been not studied because there was no real definition of what actually consciousness is. And nowadays there is a certain consensus in the field that when we say that somebody is conscious, we imply essentially that this individual is capable of having an experience, or in other words, is sentient. And when we talk about experience, we refer to simple type of perception, seeing an image, having a tactile experience, but also we may refer to more complex phenomena such as dreaming, or even having a sense of the self or the environment. And as you can see, because being conscious refers to something that is subjective and inherently inaccessible, it still remains quite challenging to know and to measure quantitatively whether an individual is conscious or not. And the current paradigm that we mostly rely on to assess whether there is any conscious content is through some form of responsiveness. So in the lab, we ask a participant to respond through a button press, to report whether an image has been consciously perceived. But also in a clinical setting, we may ask a patient affected by disorder of consciousness to react to some instructions, for example, through an eye blink or through a motor response. Yet, this type of paradigm has an inherent limitation, and this is due to the fact that a lack of responsiveness does not inform us on the absence of consciousness, because this may simply due to limited sensitivity in the way we actually assess it. And so one main motivation behind this quest for identifying preserved brain functions in the absence of consciousness is linked to this need to overcome this current paradigm, trying to assess both conscious and unconscious states based on experimental evidence, and specifically based on the neural correlates of brain functions that can occur both in unconscious and conscious states. And critically, these brain functions do not need to require some specific form of explicit overt response, because this way we can assess or measure their neural correlates in both unconscious and conscious states without any explicit report, which is affected by this limitation. And in this type of research, in the last years, we had this opportunity to measure brain activity in a specific cohort of comatose patients. This has been already introduced by Diego yesterday. These are comatose patients that are in intensive care units following a cardiac arrest. They are in an unconscious state at least during the first 2 days after coma onset. And also in this setting, they are typically managed through a quite standardized protocol, which includes targeted temperature management. So body temperature is lowered to a target temperature between 33 and 35 degrees. They're also mildly sedated. And then the day after, they are brought back to normal temperature. What is important to know here is that during this first acute phase of coma, it's quite difficult to predict whether the patients will wake up, a chance of awakening. And also these patients are all treated in a very standardized way. So they represent a quite homogeneous cohort of comatose patients. And also because they're in coma following this lack of oxygen due to cardiac arrest, and the degeneration over time occurs in the brain structures over scales of days of weeks. During this acute phase, they represent a quite homogeneous cohort that we can access quite easily in the intensive care. And the interest to measure brain functions in these patients is along two different research directions. So one is the development of early prognostic markers to inform about the chances of waking up in the long run, but also, as I said, they represent a good model for testing brain functions in the absence of consciousness in a quite uniform cohort. So when we started a long time ago, so here is these are hospital sites where we record the data. We have roughly one patient per week, at least in Lausanne and in Bern, is extremely prevalent as a etiology for the admission in the intensive care units. And this allows us also to record quite many data sets. So when we started a long time ago, we decided to test something that is quite simple, so the neural responses to classic type of mismatch negativity paradigm, where we administer sounds that follow a certain regular pattern, and this regular pattern can be interrupted by sounds that deviate with respect to speech or duration, for example. And when we started, the main view was that only patients with good chances of recovery would have exhibited at the neural level some form of preserved regularity encoding. Yet, this type of finding had been observed in patient recording at variable latencies after coma onset, and therefore this evidence might be also influenced by the passage of time. So we decided to measure this neural response to auditory irregularities systematically during the first and second day after coma onset in the same patients. And then because of the high heterogeneity in these neural responses in these patients, we assess whether there was regularity encoding at the EEG level using multivariate decoding analysis, classifying single trial EEG in response to standard and deviant sounds at the level of each single recording. So what you will see in the following is the results of this decoding analysis. We take as a proxy of the auditory response the accuracy of the decoding in response to different sound type. And also we collect the outcome of these patients based on standardized clinical score, and we dichotomize it as favorable if the patients wake up without major neurological consequences, or unfavorable otherwise. And without going into too much details, essentially what we observed is that no matter whether patients would have survived or not at 3 months, the results observed on the first and second day after coma onset show that most of the patients that we recorded could encode the regularity. We had significant decoding accuracy with respect to different sound type, and no matter whether we record them on the first or the second day, and irrespective of their long-term outcome. So this study shows that this preserved neural encoding of this auditory regularity during the first days is not really informative of whether the patients will wake up or not. But what this study instead allows us to observe is that the evolution over time of this auditory response, instead it's more predictive of the long-term outcome. And specifically, we found that the patients who would not wake up at 3 months were those that typically got worse between the first and the second day. And this is what you see here. Each point represents a patient, and the difference between this auditory response between the second and the first day. And you see that the majority of patients that do not wake up are those who get worse between the first and the second day. This allows us to identify very early on patients with high chances of having favorable outcome. And so this first study showed us not only that we can complement the current clinical assessment in these patients and provide very early information about the long-term outcome of these patients using this type of paradigm, but also that very likely if we look at very early on after the coma onset, we can identify brain functions that are preserved even in these deep unconscious states that have been overlooked so far. And so we decided based on this to look at more challenging type of auditory paradigm, and specifically the very popular local global test. For those of you who don't know this, this has been introduced more than 10 years ago by Bekenstein, but then has been really tested by many groups worldwide in different type of disorder of consciousness, but also in sleep and also in infants. Essentially, this paradigm allows to test the encoding of regularity established over the repetition of groups of sounds. Here in this example, the small rectangle represents a single tone of short duration. The big rectangle represents a long duration tone. And the idea is that by repeating the groups of sound identically over and over again, we elicit an expectation that these groups of sound will be repeated in the future. And then by presenting groups of sound that differ with respect to the last sound of the group, we study both the error that is detected following the presentation of a group of sound that is not expected, but also we may also look at the neural response to the regularity that is established within the group of sounds, so the local effect. So why this experimental protocol has been studied by so many different groups? Well, it's a very probably smart way to look at whether the brain can form a memory trace of this group of sound, and whether this memory trace also correspond or is parallel by a conscious report of this global structure. And indeed, in healthy participants, what has been repeatedly shown by looking at the auditory evoked potential at average level is that the detection of this global structure, which is explicitly reported by the participant, is accompanied by components that occur quite late after the auditory onset, spanning 300, 400 latencies after the sound presentation. Is also accompanied by the recruitment of multiple brain regions across a large network in the cortical networks, and also is one of the proposed markers to trying to detect whether a patient or a participant indeed can consciously perceive external stimuli. But the majority of the studies that had been performed up to now were looking at the sensitivity of this type of test. So how likely it is to detect this type of components in patients that we think being actually conscious. Yet nobody had really looked at how specific is this test. In other words, is it possible to find the global effect also in individuals that we think might be completely unconscious and unaware, therefore, to detect these global structures in the sequence. And that's what was our main motivation for these studies, where we consider this local global type of paradigm, first in a group of healthy participants, age matched to the classic cohort of comatose patients after cardiac arrest, so after 60-year-old. You see here an exemplar auditory evoked potential in healthy participant, where we recovered the classic N100 response to each sound in the sequence, but also the differential activity that is elicited by the response to global standard versus global deviant, which span quite late latencies, as has been reported in the literature. But what happened when we test the same type of paradigm in comatose patients? Well, here you see two examples of these auditory evoked potentials. You see that these responses are way less stereotypical. Actually, there is a quite heterogeneity in these responses. Here you see two examples of differential activity that occurs at late latencies, similar to what we observe in healthy participants, but in fact, the response at the single participant level can vary quite a lot. So also in such case, we relied on multivariate decoding analysis that does not imply any specific type of stereotypical waveforms in response to auditory stimuli. And what we found, as you can see summarized here in this graph, is that very similarly to what we observed based on simple mismatch negativity paradigm, also in this case, we found quite high decoding accuracies, both in favorable and unfavorable outcome patients that were recorded either on the first or on the second day. When looking at the significance of each single recording level, these results were significant in 10 out of 24 patients who exhibited this global effect. And now I have to say, because this study has raised a debate in the literature, we are now trying to replicate this study. So this is done in a framework of pre-register reports, which has received in principle acceptance. We are now currently reacquiring the data in another cohort of 30 patients. And we are also acquiring the standardized clinical score assessing the residual level of consciousness next to the recordings. And this data will be available for the community that hopefully will clarify whether this global effect is really linked to consciousness. So as a first summary, we show that patients in acute coma can exhibit sensitivity to the statistical regularity, both at the level of single sound repetition, but also using more complex type of sequences, and that probably this global effect does not always require conscious access to sounds. But now, based on these past studies that we carried out, we started to more recently move on trying to also incorporate information from interoceptive signals to see whether this monitoring of specific cardiac or respiratory activity can also contribute to our understanding of how the brain, in this altered state of consciousness, can process external sensory regularity. And inspiration in this context for this type of studies comes from this observation that while we perceive our environment, we are awake and interacting, our brain keeps monitoring these bodily signals in a very automatic and implicit way. And as has been already discussed by many of the speakers before me, there might be some form of competition between the neural resources that are dedicated to processing environmental or interoceptive signals versus bodily signals that are constantly monitored for survival homeostatic purposes. But then when we fall asleep or we fall into a coma following traumatic brain injury, I think that everybody will agree that we lose some degree of responsiveness to the environment. We also are less able to process sensory stimuli, although we know based on our studies on that of our groups, that we keep maintaining some level of rudimental processing of sensory stimuli and specifically sequences and regularity in the environment. So when we go through conscious and conscious states, there is a shift in the balance between the neural resources that are dedicated to the environmental stimuli and those that are dedicated to process what comes from the body. And if you remember one of my first slides, looking at this idea of trying to characterize conscious and unconscious states based on the neural correlates of functions that are preserved along this axis, then we thought that characterizing this interplay between interoceptive and exteroceptive signals could be a key to define this delineation between what is conscious and what is unconscious, both based on some form of explicit experimental evidence, but without any need of overt response. And how do we investigate this? Well, I hope you remember what Andrea presented yesterday. She already explained some of our experimental paradigms. In a nutshell, we look at how the human brain responds to different stimuli, in this case sounds, that can co-occur with the ongoing cardiac rhythm, or we compare this type of responses in a condition where the sounds do not bear any specific relation with the ongoing cardiac rhythm. And we study this in wakefulness and across vigilant states and also in altered state of consciousness, or coma. So in practice, what we do, we record the ECG of the single participant, and then online we detect the R peak of the ECG, and we administer sounds as fast as possible at a fixed latency with respect to an ongoing R peak of the ECG. And then in our control condition, we use the ECG of the same participant, but this time the ECG is recorded over a different temporal period with respect to when we actually administer the sounds. And this is crucial because this type of experimental manipulation allows us to naturally vary the time interval between R peak and sound onset while keeping a similar pseudo-regularity in the sequence. And then we make the hypothesis that if the brain has encoded this temporal contingency between R peak and sound, then this should manifest when we violate the expectation of the sound. So we omit them 20% of the time in an unpredicted way. And we make the hypothesis that in the synchronous condition, the violation will arise not just by the fact that the sound was not presented, but that the sound was not presented at the time that was expected based on this contingency between R peak and sound. And in our control condition, we omit the sound, but in this case, there is no cue that the brain can utilize to predict when the sound is coming. And so this type of experimental manipulation was used to investigate this type of cardio audio encoding in different cohorts. And this is the work of Andrea Pellentritu that acquired data in healthy participants across a wakefulness and a sleep session. We also acquired data in comatose patients after cardiac arrest. And we use exactly the same type of EEG, and we also included a third auditory sequence where this time the sound-to-sound interval is fixed, but it's matched to the average heartbeat of the single participant. And we also have a baseline condition without any auditory stimulation. So first I will show you the results that we found in sleep and then in coma. So first of all, not surprisingly, when we receive sound while we are asleep or awake, the brain processes them. And you can see this here. This is nothing new. You can reliably measure evoked activity in the different stages of sleep. And what you can observe that in sleep, and especially in N2 and N3, where we are in deep sleep, this auditory response lasts much longer than when we are awake. So then what happened when we look at the responses not to the sounds during the sequence, but during the omission? So what I'm showing you here is the omission evoked response in the synchronous and in the control condition that, not surprisingly, contain a huge component of the artifact due to the heartbeat rhythm. Yet this comparison is, we believed, well controlled by the fact that the ECG of the individual does not change across the conditions. And what you see here is the results of this comparison based on cluster permutation statistic. There are two things to observe here. One is the number of electrodes that exhibit significant difference in time, and the other is the waveforms extracted across the electrodes, providing the significant difference over time, both showing that the omission of the expected sound in the synchronous condition elicit differential activity at latencies that resemble that of mismatch negativity, so very early on in wakefulness. And this also replicate in other studies that we published a few years ago using very similar type of patterning. But crucially, when we look at the same comparison this time in a sequence where there was no specific irregularity to the baseline when there was no sound, we found no such differential activity, suggesting that this pseudo-regularity in the sequence is sufficient to suppress this error signal lock to the R peak within the omission period in the control condition. And therefore, this and other control analysis that I won't show you in detail for the sake of time allow us to interpret these results as a consequence of this encoded contingency between cardiac and auditory signals that the brain possibly utilize to anticipate upcoming sound in the sequence. Then what happened in exactly the same cohort, this time during sleep? Well, also in this case, by looking at exactly the same type of analysis, we found sustained differential response in N2 sleep of this omission-evoked activity that was not present when we look at this control analysis. Again, this suggests that even when we sleep, the brain can encode these two stimuli as coming together, and this signature occurs much later than what we observed during wakefulness, potentially due to this evidence that auditory response during sleep takes much longer than during awake. And also classic type of mismatch negativity responses has been shown also to occur at quite late latencies. But then what happened when we look at exactly the same type of responses, but during coma, this time we group all of them together based on their outcome at 3 months. These are data that are recorded on the first day after cardiac arrest. Well, we found in this case also a sustained differential activity to this omission responses that was not present in the control condition, but yet this time they unfold along two time intervals, one resembling what we have found in sleep, and one that we actually interpret as a consequence of an anticipation of upcoming sound. How can we interpret these early differences in terms of anticipation? Well, we compare within the same sequence, within the synchronous condition, the narrow response to the auditory stimuli versus the omission, and you see that in such case, this early effect disappears, suggesting that this early component is built along the sequence in order to anticipate upcoming sounds, possibly due to this integration between cardiac and auditory stimuli. We did not find such an effect in the cohort of patients that did not wake up at 3 months. To look into the single patient type of results and to try to see whether we can actually use this information also to relate this integration of cardio-audio integration to the long-term outcome, we also look at the single response. Each single dot here represents the decoding accuracy in classifying omission to in the synchronous versus the baseline. And you see here that the patients that show significant results with the field dot are mostly those with favorable outcome and also provide higher decoding accuracy, not surprisingly. We also try to look at whether this type of integration is informative about the clinical state of these patients. As I said, they are in a deep, profound unconscious states based on standardized clinical scores, such as, for example, here, the 4 score, where the threshold of 8 provides an indication that these patients are indeed unconscious. Yet within this cohort, there is still quite some variability in residual level of consciousness, if we can talk about consciousness in this case. And so we looked back at the scores that each single patient provided, and we found a mild correlation between the extent of this integration between cardiac and auditory stimuli and this degree of clinical severity during this first day after coma onset, suggesting that possibly can be also informative about consciousness levels in disorder of consciousness patients. This decoding was also applied by Andrea in participants that were awake, and you see that here there is a nice gradient of this single decoding accuracy in trying to quantify this cardio-audio integration between wakefulness and the cohort of comatose patients that are unconscious. And finally, the last result that I would like to show you is based on a much simpler metric. We used here only the readout provided by the ECG to try to evaluate whether there is this integration of cardio-audio stimuli. The results I'm going to show you in the next slide are simply based on the time interval between consecutive R peak, just before the omission, during the omission, and after, in the different auditory sequences that we utilized. And I hope you can see here that only in the synchronous condition and across all these different vigilance state and in comatose patients with favorable outcome, this omission of expected sound elicited a long-lasting deceleration of the cardiac rhythm, which was not observed in the other auditory conditions. And crucially, this was absent only in the cohort of patients where we did not find also the electrophysiological evidence of this cardio-audio integration. Now, these results, I think that for me the most interesting message is that they represent possibly the most convincing evidence that there is indeed cardio-audio integration and provide a simple way to compare the extent of this integration across different varieties of conscious and unconscious states, but also open the avenue for different type of investigation and different possible interpretation, two of which I present you here. So it is possible that the omission of sounds in the synchronous condition represent a quite arousing type of stimulus that elicit a startle response that we measure here through this deceleration. This type of interpretation is due to the fact that we don't observe such a deceleration in the regular condition, where expectancy should be even more obvious. It is also possible that this heart rate is a consequence of a top-down signal where the brain tries to teach the heart to readapt to a possible new sequence, new regularity, in order to adjust following the omission of the expected sounds. Yet this interpretation is also not conclusive, because these sounds occur 20% of the time, and you can imagine that during the night, after a while, you also adapt to the occurrence of the omissions. Yet these responses are very persistent across the whole night. There are also many limitations that I'm happy to discuss later on, this evidence, but we are currently working on trying to having a more comprehensive investigation of what underlies it. So as a summary of this last part, I show you that the brain implicit monitor this temporality between heartbeat and sounds, and this may support prediction of upcoming events. This coupling manifests also in unconscious states and with distinct features in the neural correlates that possibly can help us to identify whether somebody is conscious or not based on neural correlates of these preserved functions. And also I introduced a marker just based on the heartbeat signals, which is extremely simple to measure and that allow us to really compare on the same foot the extent of this integration. That is more convenient, for example, if we think that the EEG is also affected by differences in the background activity when we compare sleep and wakefulness, while this is not the case for the ECG. And the future direction, so we are currently investigating what is the behavioral relevance of this cardio-audio coupling in ecological type of experiments. We are trying to look at the effect of multiple bodily signals. So is this cardio-audio integration due to an indirect effect that we measure due to the coupling between the respiratory and cardio activity? And also we are looking at the neural underpinning of this integration. This is the work of Florian that presented yesterday, who is also implementing this type of experiments in epileptic patients with intracranial recordings. And why all this is important, I don't think I had to convince you, but I think this type of manipulation are really very much tuned to, trying to find this specific type of synchronicity between bodily and sensory stimuli, which are not really what happens in our daily environment or daily life, but this helps us to also try to find windows of optimal integration that we can also exploit in a more ecological environment. I'm also very keen on trying to use this type of correlates to trying to define an implicit mark of interoception. So we're trying to look at whether the extent of this integration can also inform us about how sensitive we are about our internal rhythm. And this can also, in the long run, help to identify targets of intervention in populations with interoceptive dysfunctions. And with this, I would like to thank all the people who collaborate with us and who did the work, and the funding bodies. Thank you.
Okay, thank you, Marzia. Any questions? Yeah.
Thank you. Fascinating results. I wonder why you chose the auditory modality rather than, say, visual or sensory, just because it works. And related, can you speculate on the behavioral or ecological stuff you talked about?
So for the auditory, the answer is very simple. The auditory modality is what is easier to use in comatose patients and in sleep. You can really tune the volume of the sounds in a way that you don't wake up the participants, while it's not easy to do it with tactile stimulation or even less with visual stimuli. And with the ecological design, where this is the work of one of our PhD students, Sergi Quadra, and so he's trying to gamify the presentation of visual stimuli, where the target is a fluid that can be collected or not collected, and the mask or the difficulty of this task is related to the presence of clouds and fogs, and trying to make really engaging, because in the literature there is quite a lot of divergent results in the impact of this cardiac stimuli on our behavior, in this case perceptual decision task, and we're trying to see whether if we make it more ecological or engaging, this effect become more clear, and we are trying also to make it, to tune the difficulty to see whether these effects are more prominent when we actually need to take advantage of these cues from the body in a challenging condition versus where it's obvious what we need to do, but it's really preliminary. Yeah.
Thanks for the talk. I haven't fully formed the question, but let's see where we go. So, especially the last part I liked, when there are these adaptations of also the heart rhythm relative to the auditory stimulation, and he points towards somewhat of an endogenous oscillator that tries to dynamically accelerate and decelerate according to the auditory stimulus, but it's not in comatose patients. And so I was wondering, is there a way of like predicting these dynamics of synchronizations towards predicted auditory stimuli based on the evoked activities in the brain, or like, do you already find any causal relationship?
So, first of all, we do found it in comatose patients, but only those with favorable outcome, not in those with unfavorable outcome. Yeah, so also this unconscious state. So this relates to the presentation, the work that Andrea presented yesterday. So I'm not sure this answers your question, but what we're trying to do is to see whether this effect of omission are also accompanied by a synchronization between the sounds and the underlying oscillatory activity in the different regularity types. And we're doing this first, but is not limited to, but first in a condition where the oscillatory activity is very evident. So in deep sleep, we know that the background activity is dominated by these low oscillations. And we found in two different paradigms, with slightly different experimental paradigm, but we found it, and Andrea presented some of these results yesterday, that indeed this low oscillation reorganize based on this type of regularity, and specifically, on average, the peak of the slow oscillation tend to get closer to the expected sound only when there is a regularity. So I don't know if this answers your question. Now, we haven't really linked explicitly this oscillatory results to the evoked activity. If you have ideas how to do this, I would be happy to hear. But certainly, we found that they are both concomitant.
Thanks.
Yeah.
Thank you, Marzia. That's very interesting, but of course, it's also very methodologically challenging, right? But I don't know, but it's really hard. Well, this looks to me also like a task to define, in agreement with yesterday talks, a minimal self, or test for kind of minimal self, but minimal consciousness. So in this case, I'm just thinking whether the best tests are tests which are kind of potentially revealing traces of consciousness, or whether even some basic network properties can be even more predictive, you know? Because that could be not only when you test it, but also when you look at the underground neural activity, which could be predictive. So like thinking in terms of this minimal self. So if you would run on the same patients, global versus local, and this regularity thing, then do you think that these two tests would be similar in outcome, or whether they would be differential? And if they would be differential, then it would mean that we are not really tapping into the same thing. And have you thought of really combining them?
Yes. So it's an interesting question, it's an interesting idea. We don't have the same patients that are tested with the two, and actually this is going to be quite difficult because these tests take quite a long time. But what we know is that while the cardio audio and what we see here is quite prevalent, even in patients that with unfavorable outcomes. So the majority of patients, you see the field dot has significant cardio audio integration. This is not the case for the global. In the global, we found it in less than half, but in different participants. So it would be interesting to see them in parallel and see whether they tap on the same type of network indeed. Now about this minimum self, I don't know if you are referring to these studies where we look at how the response to cardiac stimuli inform about the subjective perspective on incoming stimuli. If you're referring to...
Also exteroception, I'm just figuratively kind of refer to this. It's not identical, but
No.
It's a similar kind of, yeah.
It's similar, yes. It is possible that this type of cardio-audio integration also relate to some level of sense of your subjective perspective. I have to say, in sleep, there is current studies looking at within sleep the difference between when subjects dream versus they don't dream. So dream is still considered as a conscious experience, and there within the same state you can compare this type of influence of cardiac rhythm into your conscious perception, if you can talk about conscious perception, possibly a sense of the self. But in coma, we had to rely on these clinical scores to see whether there is any form of residual level of consciousness, but we cannot really have more specific information about what is the level of consciousness about the self or the environment. This is well beyond what we can actually measure.
Thank you.
Yeah.
And Diego?
Great talk, Marzia. I have it's kind of tricky question, I think.
Please go ahead.
Even for me to formulate it. So, have you been able to try to make sense or triangulate this kind of result with slow waves, considering that slow waves typically occur more in the very bad state patients, but at the same time, patients that are not so bad also have slow waves, and when you do event-related potentials, when you have too many slow waves, you it's hidden behind these slow waves because of the latency. And then we know that with the slow waves happens this that Andrea showed, that we have more heartbeats in the peak. Mm-hmm. So I don't know, have you tried to combine all of these to try to make sense of what is really happening. So with the slow waves, the heartbeats, and the responses to this cardio audio stimulation.
So we are trying. I have to say, so there are two comments on this, so I don't have a conclusive answer.
Yeah.
So one thing is that it's true that in comatose patients we may also find this kind of rhythm, but in not all of them. And actually they are very different in terms of the main frequency of the background activity. Yet we find quite striking results at group level and at single patient level, so meaning that this is not trivially linked to organization of one specific frequency. For sleep, we may indeed try to link these two phenomena in a more formal way. We haven't done it yet, but one main idea is to try to see whether the evoked activity is due to organization of this background slow activity, due to phase resetting, or to based on the additive model, and trying to link these two. We haven't done it yet, but it is possible, and it's certainly something that will be interesting to come up with. That being said, it's also possible that there are two different things, right? Because the locking between the heartbeat and the slow wave is really some phenomenon that occurs across the whole sequence. It's global, and we find both in the case where there are omissions and in the case where there are no omissions, what Andrea presented yesterday in two different cohorts and experiments. While the omission response is really something that is due to the occurrence of very specific events that are not possibly, driven by this underlying locking. Maybe there are two different things happening, or more than two, very likely.
Yeah, thank you.
Okay, maybe one more question.
Very interesting talk. So I have genuine question. So you told us basically that it's a matter of integration and prediction with this paradigm. So you have baseline measurements. Since we have this idea of facilitation, competition of internal and external signals, would you think that would be a good way to see how much patient responds to, for example, internal stimulation? For example, if the threshold in detection heartbeat evoked potential, it's higher or lower in favorable and unfavorable patient, if they have different threshold in detection auditory signals, if you think might be a way to see it, if it's a matter of integration of the two, if you have any guess. And then last question, a bit tricky one, if you think to a way to improve this response to this, which modality do you think that would be the more, promising in improve this ability?
Yeah.
So I don't know if the question is confused. But
Maybe I ask you what you mean with detection, threshold for detection of sounds, and because in coma they are unresponsive, right? We cannot measure behaviorally whether they detect the sound or not.
Yeah.
We can
At
have
the
yes.
neural level. So if
we
you have
have
a way to see if they are more aware about the heartbeat or this, if you do heartbeat evoked potential between
So
the
we
two
do
groups.
heartbeat evoked potential, we do auditory evoked potentials. These are things that can be measured easily, but there is not an obvious relation between the extent of this integration and the amplitude that is evoked by these different.
So it's not a way to distinguish the two group of patients.
We didn't try to distinguish, no. So the auditory evoked potential is not informative about their outcome per se. Even the classic type of mismatching activity, so the differential response to different sounds, is not really different. There are evidence of discriminative effect both in favorable and unfavorable outcome patients. So this goes beyond this type of neural response. It's something more challenging for the patient. Now, whether this depends on the extent of the auditory potential, we haven't, the strength of the response, we haven't looked at it. But what I can say is that certainly depend on the extent of the variability of the cardiac rhythm. So we know that these patients with favorable outcome also have more variable cardiac rhythm. And so these results somehow are also influenced by that, because the difference between different conditions are also due by the difference in cardiac variability. So you can take advantage of something that vary over time to predict better than if it's something regular, right? Because in such a case, there would be no difference between the synchronous and the control condition. Yes, I would say that is not linked to the extent of the auditory evoked potential based on our previous studies, but we haven't looked specifically here, this relation. Now, in terms of how to improve it, that's a very good question. So I think one direction could be really to try to better understand what is the regions that are involved in this coupling, and then possibly stimulate these regions and trying to see whether this can bring an improvement, but it's totally speculative at this point. If you have ideas, that would be great.
Okay, thank you.